Hello everyone, my name is Sarah and I'm the Engagement Manager at Cardiff University School of Medicine. Welcome to this webinar highlighting cancer research progress made during the COVID-19 pandemic. It is wonderful to have you join us and for us to be able to share with you some of the fantastic research that is progressing during these very challenging times. To begin with, I'm hoping that the majority of you have access to a second device, ideally a phone or second tablet, to be able to visit www.menti.com. And hopefully you can see now on the screen, if you go to www.menti.com, and use the code 712675, you should see the first question, where are you joining us from today? Don't worry if you don't have a second device, as we will send a link to this survey in a follow-up email after the session has ended for you to input your views. So yes, great. Everyone can now see the question and we're getting some results in as we speak. So it's great to see that there's some from Cardiff, rest of Wales, England. It's great to have you join us. Welcome everyone. I'm just going to leave it go a few more seconds to see if we get more input into this question before we move on to the next question. Hope everyone's having a lovely day. We're so excited to be doing this webinar and it's great that you could take the time out to join us. Lovely, so a few more from Cardiff and a few more from rest of Wales and England. Lovely. Okay, I'm going to move on now to the next question, which hopefully should see. Oh, we have one from the rest of the world. Welcome. Okay, now this question, just wanted to ask, before we started the main body of the session, in your opinion, how much do you feel COVID-19 has impacted on cancer research? So the options are a great deal, a moderate amount, undecided, a little bit, or not at all. So I can see from your responses so far that many of you do think that obviously the pandemic has impacted on cancer research. And that's great. I'll just leave it a few more seconds so anybody else can input into this question. That would be great. Okay, thank you. Thank you for your input into those questions. We now are going to hear about some exciting research that has been progressing in the Cardiff University School of Medicine during lockdown. During this online session, you will be able to ask questions, live questions in a written format. So please take this opportunity. You can do this through the Q&A function on Zoom, which should be available at the bottom of your screen if you're using a laptop or tablet. If you're on a mobile device, the Q&A function can be found at the top of your screen. If you like the look of a question already asked, please use the thumbs up button to promote the question as we will prioritize questions with the most likes. But we will try our best to answer the majority of your questions. Right, now enough of me. Now over to Professor Andrew Godkin, who is chairing today's discussions.
Hello, good morning or good afternoon, everyone. And thank you very much for those of you who have signed into this webinar, which overall is trying to offer a reassurance to people who are interested in cancer and the research into cancer that despite these challenging times, things have continued. We've all learned a new vocabulary in the last 4 or 5 months because of this virus, SARS-CoV-2 or COVID-19. It's been, obviously it's a pandemic which the likes of which most of us haven't lived through, and it's dominated our lives really, both in the media and online. And so I think are getting a slightly distorted opinion perhaps about what's going on. It is challenging. We're learning a new vocabulary, lockdowns, social isolation, social distancing, wearing masks in public, the kind of images that we've only seen when we've been abroad. But it's easy to think that everything has frozen and nothing's happening. And indeed, we know that hospitals have been rejigged so that to allow patients who came in, and we had a small number that came in April, but it's been very manageable. And we know the university has taken steps to protect its staff from the possibility of transmitting to each other. So access has been more limited, no doubt about that. I think we'd all agree there's been some benefits in as far as society is concerned though. You know, we experienced a period where we had less pollution, cleaner air, more time to maybe reassess our relationships with our families and friends. So this has been good. One of the other obvious benefits we've had is learning to work differently. I run a lab with Professor Owen Gallimore, who you'll hear from later on at the end. And our research drive is mainly focused on cancer immunology. We have a period in the world where we've had almost a paradigm shift in the way we treat cancers, in that we can make the immune system be activated in order to attack cancer. And you're going to hear from 2 people after me from the lab. We're going to tell them a little bit about how one of them has managed to continue a project which has been approved by the university to continue through on the bench, and also a PhD student who's done an enormous amount of work whilst working from home. So our sort of theme is to take the lab is to look at very basic mechanisms of how cancer grows and how the immune system can be made to attack it, and why it doesn't do that more successfully. So we're interested in modulating the immune system to making vaccines, and we've run a trial in fact with a vaccine a couple of years ago. And we focus on colorectal cancer, although we're about to broaden our remit to include other solid organ tumours such as breast cancer. We'd also like to take just an opportunity when we're talking about our lab functions. We're very reliant on some fantastic colleagues, pathologists, surgeons, oncologists who help us keep this pipeline going into research into human disease. So the question is then, so what have we done? It hasn't been at all doom and gloom, despite what we might, impressions we get from the media. We have a lab of around 13 people. So firstly, while sitting on this webinar, Zoom webinar, we've had endless Zooms. Actually, that's enabled us to keep very connected with our technicians, PhD students, and postdocs. And we sort of divided them into teams, those who work on particular areas. And this has been very fruitful. And indeed, we've done this with our collaborators. We've been having some Zoom meetings with people, whether it be in Glasgow or London or abroad. And this has worked well. I have to say, in fact, we've almost sort of been more connected by doing this and kept on top of things. So although, endless Zooms, it's had a huge benefit to us for research. And COVID has driven this, because, this is the way that we've learned to work. It's been safer. I'm sure this will alter in the autumn. The other thing it's enabled us to do is consolidate our research. We are funded in the cancer side of research. We have funders from CR UK, CR Wales and the Wellcome Trust. And we've really been able to sort of look at the groups who work on these areas and to write up the results. This is important. We've submitted and had accepted 3 papers during lockdown and 2 reviews. And this enables us to think about where we're going to next. It's very important that we put our work out there in the public domain to be peer reviewed and scrutinised. But the comments that come back from this process are enormously useful for us for directing our future research. In a similar way, we've written quite a number of grants during lockdown and got ethical approval for various projects, like a new project looking more at radiotherapy in human tumours. And this is a really exciting sort of area we're going to move into, both in Cardiff, collaborating with Lindra, and also collaborating with colleagues in London. So applying for grants is also a good process of scrutinising your work. Your grants get criticised and we get feedback, and this is enormously useful for us. It's often quite an iterative process, you know, toing and froing until you get a body of research which everyone thinks will be productive. And we've been able to, as I mentioned, you hear from Dr. Lauder, Dr. Sarah Lauder in a minute. We've had permission to do some what was considered important bench work to continue. We've continued a programme of work with approval from the university on essential work on laboratory-based work. And we've also had our students at home sort of writing reviews and consolidating their results. So I think it's been a strange time, there's no doubt about that, less human-to-human contact, but a lot of sort of verbal contact through Zoom and contact through writing things up and discussing these. And so we've actually found it a period of quite intense work, it's fair to say. Really, we've only followed very few people who really couldn't play a role at the moment, say the sort of technicians. Most people have been sort of onwards and upwards, and it's been great to see how people have responded to this. So I think at that stage, the final comment I'd say is that this whole period has enabled us to alter our strategy for the future. We want to exploit our research. We want to develop new treatments and explore new avenues of treatment for cancer. We'd like to explore new avenues of diagnosing cancer earlier. And this whole body of work that we're doing at the moment is really directed towards that. And now we can sort of consolidate and think about how strategically we will work going forwards. And I think this has been important. And the last thing to say from a university point of view, Professor Owen Gullimer has been made a theme lead at the college for cancer. I've been a theme lead for immunology, and bear in mind I said that we're very interested in cancer immunology. You can see that this is a very synergistic theme lead relationship. So at this stage, I'm going to hand on to the first sort of person from the lab to speak, who's going to talk about how he's managed to negotiate quite a lot of work, it's fair to say, in the first year of his PhD, and he hasn't let this hold him back at all. So it's called Lorenzo Capitani, and he'll be speaking to you now. Before I move on, I just see if there's any questions in particular that anyone wanted to ask me at this stage, and I'll take those. Okay, I think at this stage then we'll move on to Lorenzo.
Okay, hello everyone. I'm Lorenzo. As Andy mentioned, this is the first year of my PhD, which I started back in October. And today I'll be briefly telling you about what my PhD thesis will be about and how this all relates to the work I've done during lockdown whilst being at home. So the focus of my PhD thesis will be a protein known as LAG3. So imagine here we have a cancer cell. Now, within our immune system, we have lots of different kinds of immune cells, including regulatory T cells and effector T cells. Now, effector T cells have a lot of functions, one of which is to sort of hunt down and kill cancer cells. Then we also have regulatory T cells, whose function is instead to sort of prevent our immune system from reacting excessively and causing damage to the body, in essence, telling other immune cells when to stop. Now, this normally is a good thing. However, these regulatory T cells can often also be found within tumors themselves, where they prevent effector cells from carrying out their function. Now, observations made by one of our research associates in the lab, Dr. Martin Skur, identified that a significant proportion of regulatory cells in patients that had colorectal cancer expressed a protein on their surface known as LAG3. So this protein will be the focus of my PhD, and it's for this reason that we are interested in studying its function with the aim of eliminating these regulatory cells, because in theory this should enhance the immune response and better allow effector cells to kill tumor cells. Now, the story is not that simple, of course, because effector cells can also sometimes express LAG3 in some instances, but this is all the more reason to try and understand the function of this protein. So going into lockdown and having to work from home, we decided that it would be worth writing a review article about LAG3. So review articles are documents that compile studies for a relevant research topic into one document, summarizing the current state of understanding of the topic, highlighting recent discoveries, and also highlighting what gaps exist in the knowledge that we have on this particular topic. And they are a very useful tool for scientists like ourselves, because they can help us better plan experiments that sort of fill the gaps in this knowledge, and they can also act as a sort of guidebook. So if we, maybe don't remember exactly a fact or two, we can go look that up within review articles. Now, LAG3 is a very hot topic. There are more than 1,000 studies that have the word LAG3 in it, and currently 25 clinical trials investigating LAG3 as a target for immunotherapy. So it seemed appropriate to write a review article for LAG3. And so there have been 3 of us working on writing this article, and in essence, it's been months of sifting through articles, trying to draw parallels between studies, summarizing what we know, and highlighting what we don't. And this is sort of an ongoing process that we've almost completed. But once completed, this will definitely facilitate my thesis, and once published will hopefully do the same for other researchers who study LAG3, or perhaps even inspire researchers to start studying this interesting protein. Now, being at home was also a good time to work on my bioinformatics skills. Now, when we say bioinformatics, this is just a broad term that's used to refer to computer software skills and statistical skills that can be used to understand large sets of biological data. So a scenario in which we might use bioinformatics, say we give some patients a particular treatment, we then look at their genes and their gene expression to try and understand why some patients respond to a particular treatment whilst others don't. And this is sort of exactly what I've been doing. I've been analyzing a data set of experiments performed by Dr. Emma Jones in our lab. And during her experiments, she eliminated the regulatory cells that I was referring to earlier in the immune system of mice that had tumors. So I've been sort of trying to make sense of the data and try to understand what effect the eliminating these regulatory cells had on the immune system of mice that had these tumors. And it's, again, a long process that's still ongoing, which produces graphs that I find extremely beautiful, like the ones on screen. And the aim is to try and derive meaning from the huge amounts of data that these experiments can produce. And this is a skill set that's very, very useful for immunologists nowadays. It's sort of a must if you want to make the most of your experiments. So going forward, it will be a useful tool to have during my thesis when we come to performing these experiments that try to understand the function of LAG3, but it will also be useful as a general skill to have in the lab to analyze the data of other lab members. Now, under the umbrella term of bioinformatics, I've also been analyzing images of organoids. So what are organoids? Organoids are, in essence, 3-dimensional biological structures that represent the tissue which they are derived from. So for example, we take samples of colorectal cancer from patients and grow them in the lab to make these three-dimensional, what are known as mini guts. So they represent the colon in a Petri dish. And the aim is to, down the line, use this research tool to investigate how patient immune cells interact with the tumors, again, to come to an understanding of the function of LAG3. Now, when we come to perform these experiments, an important factor will be the ratio of immune cells to organoids that we have in our experiment. And to count organoids, we can do this manually. But one, this is very time-consuming, and two, it's very prone to error. So to improve the reliability of our experiments down the line and to streamline the process, I thought of applying a technique known as object recognition. So I have here a video of what object recognition is. And this is an example of an object recognition tool that has been trained to identify people, bags, umbrellas, cars, from a video. So I thought of applying the same concept to our organoid scenario. How do you do this? I had thankfully saved a very large amount of organoid images, and in essence, it was just sitting at the computer and putting boxes around each organoid and generating this very large data set, which alongside with some code, trained an algorithm that could identify organoids from images. So this will be a very, very useful tool when we come to performing these important experiments that I mentioned down the line that will try to sort of assess how immune cells interact with these tumor organoids. Now, the final thing I'll talk to you guys about today is a project I'm very excited about. So back in April, we submitted a project proposal to develop a computer game as a teaching tool to portray what the immune system does, how it normally fights cancer, and how researchers like ourselves and others are developing therapies which aim to manipulate the immune response to improve outcomes in cancer patients. So to briefly give you an idea of what the plot would look like, the player would take on the role of one of these effector cells that I was referring to earlier. And we aim to take the player through on a journey through the immune system, sort of just looking at the intricacies of the immune system up to a point at which the player will meet a cancer, and they sort of have to fight the cancer under the role of this effector T cell. The hope is that through the users learning biologically accurate game mechanics, we can teach players about immunology and cancer biology. So in doing so, users should learn why cancer can be difficult to treat, but also how we aim to manipulate the immune system to unleash its cancer fighting potential within patients. And I think this is an extremely relevant project because a number of immunotherapies already exist to help fight cancer. And really what we are living through now is, I think, the beginning of the immunotherapy era and cancer treatments. So I think it will be an exciting tool that I envisage will have many applications. So it could be used to help inform cancer patients and their families about what immunotherapies are and how they work. But I think it could also be used as a tool in schools to help educate kids and teenagers about the beauty and the intricacy of the immune system, and hopefully inspire the next generation of immunologists, cancer biologists, and cancer immunologists that will be driving the discoveries of tomorrow in the field of immunotherapies. So this has been my lockdown research journey, what I've managed to do from home. If anyone is interested in following my research or developments of our game, please feel free to follow me on Twitter, where I'll be posting updates. And we're also looking for people who are willing to provide feedback on beta versions, early versions of the game, or an art design. So if you or perhaps kids or grandkids are willing to provide us with this feedback, please just drop me an email at the email on screen. So that has been my lockdown journey. stop my screen share now. And I believe, yeah, we are back to Andy now for questions. Andy?
Hi, Lorenzo. Thank you very much. That's great. There's a couple of questions coming through here. One was about, does LAG3 have a role in other tumours? Are you muted now, Lorenzo?
No.
Yeah. Okay. So, do you want to comment on that?
Yeah. So we're sort of looking at LAG3 in the context of colorectal cancer, as you mentioned. But I mentioned previously that there are 25 clinical trials looking at LAG3. And this is in the context of a wide range of tumors. It's sort of gastric cancer, renal cancer, melanoma. So it's being sort of explored as a therapy in a wide variety of cancers. Yeah.
Yeah, it's it's interesting, isn't it? I know in some as a monotherapy it might not work. We'll wait and see, but I think in combination it's got
Promise.
Promise, definitely. And I think, as you mentioned, from our perspective in colorectal cancer, where we know that other checkpoint inhibitors, the drugs that have been used for melanoma, don't work. We're excited to look at LAG3. Also, we know that it has a role in breast cancer, so we'll be looking at in the future. So the other thing, Lorenzo, a question that's come through is someone saying, how difficult is it to get patient tissue into an organoid? And just before you answer, I'd say we are reliant on our colleagues in pathology and surgery to help us do this, and we've got a great sort of team like that. But Lorenzo, how have you found it?
So it's definitely not been easy, and we sort of go months where we don't have any samples that we can use to grow organoids. But in essence, as you say, we have connections with pathology, and we sort of go talk to patients who are about to have a colon resection as part of their treatment, and ask them whether they would be willing to donate a part of the colon that would normally be removed during the surgery and would therefore be classified as biological waste. And if they are happy to give us a sample of that, then we take it back into the lab, sort of mush it up, and put it into a solution that's rich in nutrients and in factors that stimulate its growth into organoids. So it's not
easy.
Well, relatively, yeah.
Okay, very good.
Is actually getting the samples.
It says there's another question here. What's the link between LAG3 and the effector regulatory T cells? I think you've probably touched on that. But we know LAG3 is expressed on cells which have a regulatory function. They're also expressed on activated cells. So there's a balancing act here between if you neutralize it or kill those cells, between killing cells which are doing some good and cells which are regulating the immune system. There's a question coming in at the ratio. What's the ratio in relation to the organoid reaction? Just looking through these questions, that's quite a few. What's so special about an organoid for research? What would you like to comment on that, Lorenzo?
Well, I guess conventionally before organoids, we would sort of be stuck to either using mouse models or using in vitro models that are sort of done on a 2D surface, and that's not a very good representation of tumors, within patients, because these obviously have three-dimensional structures that are a lot more complex than conventional two-dimensional models. So organoids have the advantage, first of all, that they are three-dimensional, but also because they grow from stem cells So in our case, from stem cells that are present within colon tissue, then the resulting organoid has a lot of different types of cells. And this is sort of thought to be a more accurate representation that we can use in vitro to study tumors.
There's another a good question come through, which is we've already sort of touched on it a little bit. It said, do you expect LAG3 inhibitors to unlock the immunogenicity of tumours considered to be cold? So a cold tumour is something which isn't very immunogenic. It's not maybe naturally full of lymphocytes. Or it says, or do you expect those tumours traditionally considered responsive to immunotherapy like lung, melanoma, renal? So our experience on this in colon cancer is that there are a lot of lymphocytes in a lot of cancers, colon cancers, and they look to have effector functions if you remove the regulation of it. So I personally think LAG3 is an excellent target. And the reason I say that, we alluded to Martin Sker's work, was that when we block these cells, you unleash quite a striking response. And they are very, very suppressive within colon cancer. So we know that anti-PD-1 and anti-CTLA-4 don't work. So I think it's an exciting prospect, but like all these things, it will probably be in combination with other immunostimulants. Right, I think in the question of time, there's loads of questions coming through. I'm just scanning through to see if we missed out on anything. I think we're going to have to move on a little bit, but at the end, we'll have another chance to put all the questions together that have cropped up. So, and I think some of the same questions might apply to Sarah's talk. So we'll move on to Dr. Sarah Lauder now, who's going to talk about her work on breast cancer in a model system. Lorenzo, thanks very much.
Bye-bye.
Okay, hi everybody. Let me just start my screen share. Okay, so I'm a postdoctoral researcher in the Gallimore Godkin group. I am funded by CI UK, and I basically work on the kind of preclinical models within the lab, really looking at how we can manipulate the immune system to try and control cancer. So both Andy and Lorenzo have touched a little bit on what we do, so I'm going to go into a bit more detail about what I've kind of done during the COVID-19 pandemic. So fortunately, I was one of the researchers that's been granted essential user access, which means that some of my projects we couldn't just stop during lockdown for kind of ethical reasons, and also they were deemed to be essential for furthering cancer research. So kind of, I would say 50% of my time has been spent at home, working from home, and 50% has been spent going into the laboratory. So during my time working from home, this has actually been really useful. We've managed to get a lot done, one of which Andy suggested, you know, we have actually managed to get papers published. So one of the papers that we had in the pipeline prior to the kind of lockdown phase starting, we had reviewers' comments back on this. We've managed to address these comments. We've managed to rewrite the paper. It's been back out for review and it has been successfully published. So that's fantastic to be able to get our research out there during this time. So that's been brilliant. Another thing that we were sort of asked to do just prior to lockdown, which has been really great, it's been something that we've been able to work on, is to share the methodology of the tumour models that we have here in Cardiff. So we are, I would say, kind of world-leading experts in using a spontaneous fibrosarcoma model to study tumour immunology. So with other lab members, we've worked together to provide a detailed description of the methods that we use for this tumour model, and this is going to be published shortly. So this will allow other researchers looking at cancer models across the globe to, rather than start from scratch and try and tweak things, they'll be able to actually use the protocols that we've put together to go straight into their cancer research. So this will further cancer research in a number of different aspects for researchers worldwide. So in terms of my essential laboratory research, obviously you know, Andy kind of suggested, you know, what we do as a lab group, we do kind of basic research, we do preclinical models, and then we go into kind of clinical trials. So in terms of our basic research, we do a lot of kind of manipulating of both immune cell subsets and tumour cell lines. We look at how these interactions work together. We analyse a lot of data as a result of this. We do a lot of thinking, a lot of reading of the literature. And then what I kind of do in my kind of aspect of the lab group is to look at this in a preclinical research setting. So this ultimately involves taking forward the findings that we've had from our basic research and look at the best ones that we can use in a tumour model. And obviously our ultimate goal is to try and cure cancer in those tumour models. So both Lorenzo and Andy have discussed this earlier. Obviously, we want to try and manipulate the immune response to try and control cancer. So T cells are something that we're interested in the lab. So if you think about it, we have a growing tumour. If we can get T cells in there that are responsive and have killing functions, then we are able to potentially control that cancer and basically clear it. So that's great, but the tumour itself obviously employs mechanisms by which the T cells can be suppressed, so they're no longer able to do this. But also we have a second arm of the T cell immune response, which we call Tregs. So these are great for controlling things like autoimmunity, but they're not so great in the context of cancer because they employ a number of suppressive mechanisms that can result in our T cells going from being responsive and able to kill the cancer and become unresponsive, which results in the tumour growing. So as a lab, we've employed a number of different strategies to try and either deplete the number of Tregs so that they therefore don't have the same numbers to try and control T cells, or to try and inhibit their suppressive mechanisms, and therefore either of these two pathways can result in an improvement in the number of responsive T cells that can get into the tumour and also the quality of their cancer killing. So ultimately we hope that we can control cancer. So obviously, we look through the literature a lot and we try and look for new methods in which we can use to try and inhibit our Tregs. And if we can repurpose drugs that are already out there, this is great because we know that they'll have already been through clinical trials, so we know what their side effects are in humans. So one of these that we came across was a PI3 kinase delta inhibitor, which is actually used for treatment for blood cancer. So in the context of this, in terms of blood cancer, this drug is taken orally, and I'm going to refer to this as PI-3065 for the rest of the talk. So that's this. We know when this is given to patients that this can actually kill the cancerous B cells within the bloodstream, and this can result in clearance of your leukemia. But what we found interesting was that Tregs can also be targeted by this PI3 kinase delta inhibitor. So we wondered if we gave this PI3 kinase inhibitor as an anti-Treg drug to tumour models, so to mice that have solid tumours, whether if we could get rid of these Treg suppressive effects. So that's basically been my project for the last few years. So if you think of the PI-3065, so this is our drug as an anti-Treg treatment, we've employed this in a breast cancer model in mice and have looked at the effect this has on the growth rate of these tumours in mice. So we have what we call vehicles, so these are basically mice that are untreated, and we look at the tumour growth rate. So this is time across the bottom compared to tumour volume. So this is a measure of tumour burden. So you can see that these tumours grow quite quickly in the mice, but if we treat them with our anti-Treg therapy, we can see that we not only slow down the tumour growth rate, but we also reduce the total burden of tumour in these animals. So when we looked at this in more detail, we found that actually animals treated with this anti-Treg therapy fall into two groups. Those that can slow their tumour growth, as shown by the white circles here, compared to those that actually have the capacity to actually completely eradicate the tumour, as shown by the grey circles here. So this made us question what was the difference in this therapy and what it did to the Tregs between animals that just control tumours and animals that are able to completely eradicate their tumour. And we find that we come back to LAG3. So we found that on our Tregs in mice that just control their tumour growth but don't eradicate the tumour, we find elevated levels of LAG3 on the surface of our Tregs compared to those that are able to eradicate the tumour. So in light of this, we thought, well, why don't we use an anti-LAG3 treatment, which obviously Andy and Lorenzo have both suggested. These are now in phase 1 early clinical trials in patients, to see whether if we use both of our Treg treatments, whether this has a combined effect. So these are our growth curves again, where we're looking at untreated animals, and we see that the tumours grow quite quickly and the tumour burden is quite high. If we treat with our first treatment, so our PI-3065, we find that the tumour growth is much slower and the tumour burden is lower. So what happens if we just give the anti-LAG3 alone in the breast cancer model? We don't really see much effect. It looks pretty similar to untreated. But if we give the two anti-Treg treatments together, we find that our tumour burden is significantly reduced. So the time course of growth is much slower, and their overall tumour burden and survival is much better. So at the start of the year, we wrote this up for publication, submitted it to a journal. And as Andy said, you know, when papers go to journals, they undergo peer review. So they're really scrutinised for the science that we put together. And then what happens is that you get comments back to try and improve your data. So literally just before lockdown happened, we had our reviewers' comments back, and that's what I've really been addressing during the lockdown phase in terms of our research. So one of the things that the reviewers asked for was, that's great that you can do it in a breast cancer model, but can you do it in a different type of tumour model? So here we've used a colorectal cell line in our mice and used the same two treatment strategies again. So we find that if we just give the LAG3 alone, which is in the white squares here, this doesn't really have an effect as a monotherapy. But if we combine it with our PI-3065, which was our first line therapy, we see a significant improvement in both the kinetics of tumour growth and the overall tumour burden, with better survival for these mice compared to just treating with the PI-3065 alone. So this demonstrated that we can do this in more than one tumour model. It's not a tumour model specific effect of using these two treatments together. Another thing that we were asked to do was to try and use these two treatments in a more therapeutic strategy. So obviously, if you're diagnosed with cancer, you will be showing symptoms, you may have a mass, you may be able to feel the lump. So with this set of experiments, what we were interested in doing was trying to treat those mice once we could see a visible and a measurable tumour, so more representative of the kind of treatment that you would have in the clinic. So these mice, once they had their tumour established, we would start treatment. So here we're looking at just tumour growth rate of the breast cancer without treatment. If we give our PI-3065, we find that this still slows tumour growth rate and improves overall survival and lowers tumour burden. How about if we give the anti-LAG3 as a single monotherapy? Well, we find again that this doesn't really make too much of a difference compared to untreated mice. But the two anti-Treg therapies now, so our PI-3065 and our anti-LAG3 given together, significantly reduces tumour burden and slows tumour growth over time. So following on from kind of what Andy and Lorenzo have both said, we've shown in both a breast cancer model and in a colon cancer model, and we have other data that we've now got coming through to suggest that actually this treatment as a dual treatment could be successful for solid cancers and warrants further study in human settings. So this is the data that I've really worked quite hard on during lockdown. It's definitely kept me busy, and it's, you know, we've now finished our reviewers' comments. We are rewriting our paper and we're due to submit shortly in the next couple of weeks, and fingers crossed this data will be published. Okay, so that's me pretty much finished, so I shall stop sharing my screen. And I'm happy to take any questions.
Hi, Sarah, thank you.
Hi.
Great. There's a question which sort of overlaps a little bit from the previous talk.
Yeah.
Again, it's about combination. And this is why I think this will be interesting in breast cancer. The question was saying, you mentioned trials looking at a range of cancers and gastric. You mentioned, can you discuss how this is aiming to research hard to treat cancers such as pancreatic cancer or gastric cancer is hard, but also with breast. And I think what the combination of facts we're trying to do is trying to make tumours what's called hot, to make them more immunogenic. I think tumours which are irradiated, like breast or sometimes pancreatic cancers can be, has a chance to alter how immune they are and allow lymphocytes in. And then this is where we think, using combination of maybe PI3K inhibitors or with anti-LAG3, etc., offers the most chance. So the questions are right. If you don't have any lymphocytes in a tumour at all, if it's very cold, then you need to do something to alter the way they get in there, either the way the blood vessels feed the tumour, or the way the tumour is sort of killed and sends out danger signals through low-dose chemo or radiotherapy to allow lymphocytes in. So I think they're really interesting, these results. The anti-LAG3 alone really replicates what we knew before, that it doesn't seem to do anything by itself, but in combination it's really interesting. There's a question coming through about using organoids from other parts of the body. We are about to start, once we have access to patients, etc., when the hospital opens up a little bit more, growing breast cancer organoids, to look at these very questions about whether we can impinge on these with a combination of either anti-LAG3 or PI3K inhibitors, or even giving something like low-dose radiation to the organoids to alter their immunogenicity. So these are one question that's come through is, do we have a lot of collateral damage when you kill the Tregs? I would say it depends how many of them you kill, but I don't know what you think about that, Sarah.
Yeah, I mean, obviously Tregs are essential for controlling autoimmunity, so there is side effects, and you do see this in patients as well when you give them kind of treatments that kind of target regulatory T cells, and we do see this in our tumor models, but the side effects aren't huge. We don't lose animals to side effects. We do see some kind of skin effects, but ultimately it's the same as you would see with patients. The trade-off is that you have some side effects, but you are able to control the cancer. And actually, once we, once the animals have eradicated the tumor, if we take them off the treatment, then with time those side effects dissipate.
The other thing, talking about depleting Tregs, is we know in animal models or in humans who you remove all the Tregs or born with defects, which means they don't develop Tregs, they do get this sort of autoimmunity endocrinopathies. And so the ways of more gently removing them, and we've used in a clinical trial very low dose cyclophosphamide, low fixed dose, to be very effective at targeting the most active regulatory cells, leaving those intact. So none of the patients get autoimmunity, but it can unleash the immune response, and this can be enough in itself just to control and really slow down the tumour, even in patients with quite advanced disease and current cancer. And we're looking to exploit this in early stage diseases where people have had curative procedures, but we know that risk coming back. I think it's a very exciting opportunity to give safe, non-toxic intervention into high-risk patients. So I could give the example of colon cancer, where when you've had a curative procedure, we know 25% will relapse over 3 years. So this is a great opportunity for us to intervene with them. And we know from the animal work as well that things like cyclophosphamide, which just take out a proportion of the Tregs, leave the animals healthy, but will control the tumours, and in animals actually eradicate them. So it's really exciting. There's a very good question come through which said, when do we think dual therapy will be available on the NHS? I suppose dual therapy here we'll be talking about in breast cancer, maybe PI3 kinase delta inhibitors with LAG3. LAG3, as we said, has already been used in a number of studies as monotherapy. For some of the reasons we alluded to in our experimental work, I think monotherapy probably won't work, but we'll wait and see. And PI3 kinase delta inhibitor has already been used in monotherapy. I think so. It's lung cancer. I forget which tumor it's used in. With some
clinical trials for that, and obviously it is a licensed therapy for leukemia, so I think it's follicular lymphoma.
So I'd be very interested in exploring setting up an early phase 1 study to look at a combination in something like breast cancer, where we've got very good preclinical data, and this is something we'd look to sort of set up going forward. I'm just looking to see if there's any, there's questions here, so I'm scanning down. I think we've done most of these now. You mentioned you had to remain in the lab due to ethical reasons. That's a great question, actually. Do you want to just talk about, we did have very good reasons for doing that, sort of, so
obviously lockdown was kind of put onto us quite quickly, and we were kind of halfway through a study with animals, and it's not ethical to just stop that study. So we applied to the university to ask them if we could carry on with that study. So obviously I went in every day and did the usual research that I would do with those animals, so that we could continue that study and generate the data from them, so that the animals weren't wasted. So that's really what we kind of mean by the ethical side of it.
There's another good question here about organoids again, saying organoids are still different from how the body works, as there are many systems interacting. Do you take this into account when researching? Well, this is one of the reasons we're really trying to exploit the system, because you're absolutely right. If we just took a tumour cell line and grew that into what's called spheroids, you wouldn't have possibly the interacting cells that we know in a tumour. We know that the background tissue, what's called the stroma, the tumour matrix, is really important for facilitating the way the immune system works, and also facilitating where the tumour grows. So I think the beauty of the organoid system is that you also have other cells which are interacting with it. And of course, we're very interested in lymphocytes. So we want to get a system where we have an organoid with the supporting cells and lymphocytes, and then we can see how to activate those. I'm just scanning down here. Sorry, the questions. I'm zooming forward. Dual treatment, sort of like that. The collateral damage, we sort of talked about that as well. So another question about targeting the LAG receptor, LAG axis at the level of the tumour, so the level of the T cell, then you may just reset the immune system locally instead of systemically. That's a good point. We're not sure. It's still unclear what cells exactly express LAG3. Obviously, it's on lymphocytes, B cells, probably on some monocytes. There's also one or two intriguing reports out this year and the beginning of last year that it may be expressed on epithelial tissue. It might also be expressed in the brain on microglia, and some of the ligands for it are in the brain. And the role, because there's very few lymphocytes in the brain, whether it plays a role in the pathology of common diseases that we're facing, not just tumours of the brain, Alzheimer's and Parkinson's, remains to be seen. So there's a lot about the biology that's unknown. We don't even know how LAG3 signals in the cell. So it's a really intriguing area to be researching into at the moment. We're also trying, in regard to that question about trying to target the tumour, we're also looking at using something called an oncolytic virus in collaboration with Dr. Alan Parker, who works in our institute. These viruses, for reasons that aren't totally clear, can target tumours, replicate in them, and kill them to a degree. And obviously, if it worked 100% effectively, that would be a treatment in its own right, but they don't, because the tumours can escape. But we're now incorporating some of the antibodies we've made, the LAG3, we're putting those antibodies expressed in the virus, so the virus can get into the tumour and kill the tumour, or some of it, but can then, before it's dead, can synthesize and release antibodies which will target LAG3 just in the tumour environment. So we think this may be a really exciting kind of dual approach. Keep talking about targeting tumours by more than one avenue so that they can't escape. So I think that's probably, oh, there's another question just come up. Someone said, we've had a few people asking things about, I'd like to research this more, can we target resources, etc. I think when Owen rounds up the end, she's going to give her email address. And you have the email addresses also of Dr. Lauder and Lorenzo, so and myself. So you're more than welcome after this. Any of you have any questions or want to explore these things further, to email any or all of us, and we can also direct you into reviews, etc., about these areas of biology that we're working on, and our own reviews. So thank you very much to everyone for those questions. There's been more than we can answer in the time allotted, and they've been really stimulating questions as well. These are really important things. When are we going to move from animal models into human studies? Can we get these human studies going? Of course, funding is always the issue in this day and age, but we apply, continue to funders, and that's really, we're well situated here to exploit our colleagues in Melindra and in the hospital to set up clinical studies. So at that point, thank you to everyone who's signed into this webinar. I've said enough. I'm going to pass on to Sarah Hatch, who's going to just look at the look at the feedback, and then at the end, Owen's just going to give a very brief cheerio. So thank you very much to everyone who's contributed this morning, and to the other speakers.
Thank you everyone for those really engaging questions. I'm now going to share my screen again, and hopefully you can go back to your second device and visit www.menti.com, and it's the same code as before, 712675. And I'm just going to re-ask the question that we asked at the beginning of the session. Now that you've heard from some of our researchers, in your opinion, now how much do you feel COVID-19 has impacted on cancer research? And we just value your input. Now you've listened to Lorenzo, Sarah, and Andy talk about the research that they've been doing during lockdown and during, obviously, the pandemic. So thank you very much for inputting your feedback into that question. That is great. There's just a few more that I'd like to put to you to get your valuable input into. Just leave it a couple of more seconds, I think, for anybody else that would like to respond to this question. So 7 of you so far think it the COVID-19 has impacted a great deal on cancer research. 12 a moderate amount, and 11 of you a little bit. Okay. Okay, so do you feel reassured that cancer research is progressing during the pandemic? So there's 3 choices here: yes, maybe, and no. Again, we just want your input into these questions, just to give us a little bit of feedback about today's session and see how we can improve them for future. So that's great. Thank you very much for your input into that question. Now we'd like to ask you why you attended today's session, and if you could select your top 2 reasons. The choices are to be informed, to be able to put my question to cancer researchers, to be inspired, to take part in a Cardiff School of Medicine webinar, and there's another choice as well. So quite a few of you joined today to be informed. And then many of you wanted to take part in a Cardiff University webinar, which is great, thank you. And then 10 of you wanted to be inspired. We hope we have done that, listening to the talks from Lorenzo and Sarah and Andy. And a couple of you then wanted to put your questions to cancer researchers. We hope that you had your question answered. I know Lorenzo and Sarah are busy typing answers in to respond to questions, but like Andy said, you're always welcome to contact the researchers, as well as CU Medic Engage at cardiff.ac.uk, and we can follow up with any questions that you have there too. We would like you to rate on a scale from 1 to 5, 1 being very dissatisfied and 5 being very satisfied, the usefulness of the discussions today, the opportunity to ask questions, the value of the session, and the opportunity to feedback. It's great to see the opportunity to ask questions was highly rated. It's a crucial element of the webinar was the function for you to be able to engage directly and put your live questions to our researchers. So we are really pleased that you valued that also. And like it's nice that you also value the opportunity to feedback, and I really value your feedback during the session, so that is fantastic. Okay, just a couple of more questions now. If you would like Cardiff University School of Medicine to organise more webinars, we just thought we'd be nice to ask you if you could indicate some topics of interest that we could follow up on. So if you do have a topic of interest, then please do let us know here. And if it's not something obviously within the School of Medicine, obviously the neuroscience, cancer genetics, gene therapy are, but there may be topics that we could hand over to our colleagues in different other parts of the university, and to show that there is interest in these topics. That's great. So more current cancer research, okay, ethics, more medical research, clinical trials in cancer research. Quite a lot of topics coming up here, so it's fantastic to see. Vaccination advancement, COVID-19 research particularly, yes, I think, yeah, that is very topical. And okay, I'm trying to move it down. There we go. Immunotherapy research. Okay, things to write in a medicine personal statement. Okay, well, this would be really useful to your report in a personal statement. Okay, so lots and lots of topics, so we will go through these and take these forward in the future. Thank you for that. So finally, from me, the last question, if you could just provide us with any additional comments, suggestions or feedback, obviously optional, and that would be fantastic. Just a big thank you from me for joining us today. If we do have, oh, thank you. That would be great. And then while you're providing feedback to this question, it's just to say really, thank you once again for joining us, and I'll leave it just a few more seconds for you to input your feedback here, and then I'll hand over to Professor Alwyn Gallimore, who will actually close the session. But thank you once again for your feedback. It's really invaluable, and I really appreciate taking the time to do this for us. As I said earlier, I will send the link to this, so anybody that didn't have a second device available to them, you'll be able to input your views into that link after the session has ended, when we send the follow-up email. But thank you again, such lovely feedback coming through now. Great speakers, and short and sweet. Yes, it's quite hard to concentrate for long periods of time on Zoom. Brilliant, thanks very much. And I'll hand over to Alwyn now.
I'm just going to Can you see my screen? Yeah. Okay, so again, I'd also, can you turn this audio off? Just a second, technical hitch. Okay, so I'd just like to thank you again for attending the webinar today. I set this up because I have heard so many times over the last few months how the prevailing belief is that everything non-COVID is either grinding to a halt or being at least put on pause. And that's very much in conflict with my own experience of seeing researchers mitigate against the restrictions imposed on us by COVID-19. And you'll have heard that there have been some positives. We've had a chance to reflect. As lab-based scientists, we're usually compelled to get into the lab and to do as many experiments as we possibly can. So just to be able to sit back, take stock of our data, reflect on what it's telling us, discuss with our colleagues, both home and away, what the meaning of these data are, means actually that we can move forward in a far more informed fashion. You'll also see that, especially from the talk of Lorenzo, that people have taken this time to learn new skills. They've gone on courses to learn about computational and bioinformatic methods. It's very difficult to take this time out otherwise, and that's actually rather counterproductive. So to have had that time given to us is almost a gift, because we will return to the lab as far better scientists, better equipped with more advanced tools that we can use to analyse our data. You've also heard from Sarah that there has been some bench science still going on. And Sarah didn't say this herself, but she and others who are in a similar situation have to do this safely, of course. And in order to do that, they do work, Sarah in particular, at hideous hours of the morning, and that's every day of the week. I receive a WhatsApp message to say who is in and out of the lab, and Sarah's is always the first to arrive. So essentially then the message of this is just to say that it would be wrong to say that things are perfect or ideal. COVID-19 has changed the way we work. It has imposed restrictions on us. But the overriding message that I hope that I can give you is that as a community of researchers, we are doing our best to mitigate against this and to do what we can, and more than before in some areas, to meet and overcome these challenges. So thank you very much again. And I wish everybody a nice rest of the day. Oh, and I should say before we go that the show goes on, as I hope I've impressed upon you. And should you have any questions that we failed to address today, then please do email me at that address. Thanks.